The last presentation of this session for the Global Food System Seed Grant activities is with Dr. Ryan Hansen. Dr. Hansen is an associate professor in chemical engineering here at Kansas State University and worked closely with Dr. Tom Platt, who is an assistant professor in biology. They will be talking about site-specific screening and development of biofertilizer formulations for enhanced yield of corn.
So thank you for the introduction, Maureen. And again, I'm going to be talking about our project, which was related to improving how we can formulate biofertilizers so that they're more reliable, specifically with respect to growing corn. So yes, this was a collaboration with me and Dr. Platt in the Division of Biology. And so to begin with just some background, I think we are aware, obviously, of the need for sustainable agriculture driven by climate change, decreasing arable land, increasing global population, and really a pressure to produce food and produce it sustainably. And of course, the drawbacks that we have with our chemical nitrogen-based fertilizers, whether we're thinking about energy costs of production, transport, and storage, or the fact that they're overused, leading to water pollution, greenhouse gas emission, poor long-term health, and so of course many drawbacks that that many of us are familiar with. Well, plant growth promoting rhizobacteria have potential to at least lessen maybe our dependence on chemical fertilizers, not necessarily replace them, but maybe allow us a lower energy, environmentally friendly and sustainable approach to food production. For those that are unfamiliar with the rhizosphere, it's basically that zone right outside the plant root that's very nutrient rich. It has what's called root exudates in it that are food for bacteria. And so it's really where a lot of the action happens for nutrient uptake and plant health. And so that's really where we want to focus on, and where if we're using microbes as biofertilizers, where we're really focused on is the rhizosphere and the endosphere, which is inside the root. And so there's a need in this area for really two things. One, finding new bacteria that have new functional capabilities in the root surface, and then also engineering that bacteria so that we have some kind of control over its function. So a little bit more on plant growth promoting rhizobacteria. They have a lot of known benefits. This is a list here. So for example, they can produce plant hormones to increase root development. A big one is nitrogen fixation. And so that's fixing nitrogen into ammonia so that the plant can use it for biosynthesis. There are other benefits as well. Again, I want to focus on nitrogen fixation in this talk, primarily because we're going to be looking at a bacteria called Azospirillum brazilens. This is a diazeotroph, which means it fixes nitrogen to ammonia and can be used to stimulate plant growth. There are a lot of drawbacks to plant growth promoting rhizobacteria and potentially negative perceptions of these types of formulations. Inefficient, unreliable, they're specific to a host, which means that they're not really broadly applicable. And translating that to producers, that means high risk. And so, of course, then many producers are hesitant for using plant growth promoting rhizobacteria because of the known risk that could be incurred with using biofertilizers. Well, there's a lot of limitations to biofertilizers. And so we're really just looking at one of them here in this project. And that's that plant growth promoting bacteria. We talk about how their benefits are going to occur when that plant functions in the rhizosphere. Well, one limitation is that these plant growth promoting rhizobacteria have poor survival and colonization into that rhizosphere. Why is that? Well, one reason, one very big reason, is that microbes don't exist in a vacuum, maybe like you see on your left, which is how we've really thought about microbes for many decades. But of course, in the last 10, 20 some years, we've got more of an appreciation of the fact that microbes are existing in what we call communities. So these are basically collections of many different types of microorganisms that are, it's not just bacteria, fungi, viruses, right? A whole collection of different microbes. And they're social creatures. And so they are interacting, and their interactions can be beneficial, they can be detrimental. But understanding them is a very, very big challenge because of the complexity that we see in these natural ecosystems. But it's important because if we want to find biofertilizers that are going to work in the rhizosphere, we have to understand how they're going to interact with what's already there. So our goal was to really find synergistic microbes that improve the survival and function of well-known biofertilizers like Azospirillum, Brazilens, and basically explore this idea of pairing Azospirillum with its synergistic members to see if that helps improve how Azospirillum survives in the rhizosphere environment. And really what's believed is that there are already many potentially useful strains already present in the rhizosphere. We just haven't discovered them yet. We haven't been able to find interactions yet that are important. So that's what we were out to do. So we've been kind of working on ideas related to that for a long time, Dr. Platt and I. And so we started working with a novel device. This goes all the way back to 2016, and when we were both kind of early in our assistant professorships here. We had this technology platform called the Microwell Recovery Array, and the idea is that we improve how quickly we can screen interactions. The way you would classically do it is a very slow method. It's a co-culture where you spot microbes together and see how they grow, potentially measuring 10, if you're really ambitious, maybe 100 interactions at a time. Well, we basically scale that down into much smaller sites called microwells, where we have these co-cultures that we can look at many co-cultures at the same time. And so we're talking about 10,000 on that order type of co-cultures. And so the idea with this platform is that we take one focal species. This is what we call it. It's something like Azospirillum brazilens. It has something that we know is important, and we want to find the interactions that are going to affect how that functions. So what we do is we take our microwell array and we mix it up with a microbiome, and we watch how the focal species, and this is a fluorescent labeled version of bacteria, how it cultures when it's around different members of a microbiome. And each one of these wells is a little bit different. So there's different pairings that are occurring here. So we're looking at actually a lot of different conditions at the same time and finding the most important interactions. So this is something, again, we've been working on a long time. It's a high density co-culture technology. It's pretty well developed at this point. We have several publications on it, as well as a patent that we just got awarded, that was just awarded this year, related to some of the materials we use in that device. And so our idea was to use that and to screen corn and the corn rhizobiomes. When I say rhizobiome, I basically mean that community associated on the roots, to find interactions that are going to be synergistic with Azospirillum. So what we do is we take Azospirillum, which would be in green here, we mix it with the microbiome. Again, the microbiome has potentially thousands of different species of bacteria in it. And we put these in little wells and we culture them, and we have image analysis tools that will identify wells where we see synergistic interactions, meaning that our focal species is going to grow and survive at higher levels than the background. And then what we can do, we've developed these methods, or we can pull those out with a novel light patterning tool in my lab, and then we can sequence them genomically and identify who those interacting pairs are. And so we can do this. Again, we've demonstrated this in papers in just last year. Actually, a very significant paper came out related to this technology in a different system. And so our project here, again, was to do this with corn, and we know Azospirillum is important in corn. It can be used to stimulate corn growth by nitrogen fixation. So can we find synergistic organisms to Azospirillum that are already in the corn rhizosphere? So that was our first objective. And so it really kind of follows that same workflow here. We are going to extract the root microbiome, or the rhizobiome, as I'll call it, combine it with azospirillum in our screening devices, find the most synergistic or promoting interactions, and then I take those out, characterize them. And then the second part, which was really where we focused a lot of work on because the platform was already developed, is that we wanted to show that what we can discover with the screening tool actually will show benefits to a plant phenotype. So can we not only find these interactions in the device, but does that translate to potentially improve plant growth? And so that's a big question. One we certainly haven't answered in full yet, but we definitely are in this project. We're out to get preliminary data showing that that was feasible. And so if we can do that, then we have a couple things. One, a very novel bioprospecting tool where we can screen environments and find important bacteria that are potentially useful in applications. And then it also goes to this idea of co-inoculating with biofertilizers that are not just one strain, but a collection of strains that are symbiotic, if you will, with each other. Okay, so the project we started, this was early on in the project in summer 2020. My graduate student, Niloy, is shown here sampling roots. So we did this through K-State Research Extension, and we were able to get corn roots both from tilled and non-tilled soil at a producer site in central Kansas. Our first step here was to characterize with genomically what we were getting as far as how diverse these samples were. So we did what's called 16S community analysis to figure out who was in the native microbiome as far as the types of the families and the genus of bacteria that were there. And then importantly, we looked at cultures because we have to culture in our device. We looked at culture media that would recover the most highest diversity of species. If you're familiar with environmental microbiology, you know that the vast majority or a lot of what you can culture of what's in an environmental microbiome, you can't culture very easily in a lab. And so we were out to really find the best media. We were actually found media, TY media that was useful, that actually recovered quite a bit of the OTUs that we saw in the original population. And so we optimized our media there so that we could recover really the most diverse amount of microorganisms present. Okay, so now on to the actual platform. Again, what we're going to do is we're going to take we're going to wash out the microbial community associated with the root, and we're going to put that in our platform with Azospirillum braziliensis, which expresses a fluorescent protein called GFP, and screen those interactions. And so the way we do this is we first of all get a baseline, and that is just the monoculture. So that means we're only putting Azospirillum in there, and we're just characterizing its growth in this environment, growth kinetics with respect to the wells and the media that we're using. And so we get a baseline curve, and then we put in our microbiome, and we obviously see a change. So what you're going to see is that the data kind of goes everywhere. It's not really that smoother trajectory, but you get a lot of areas where you see amplified growth of azo and also diminished growth. And so likely there's antagonism going on in some wells where you see the diminished growth, and in the areas with higher growth, you're seeing synergistic interactions, which is what we are looking for. And so, for example, the green data plot here contained one of the top wells that showed the most synergy, right, or the most promotion of azospirillum. So we can now take those cells out of the wells, and we did this for 2 wells and took multiple samples from the wells. So we had 6 samples out of 2 wells overall. We validate these interactions because we have to kind of have a secondary validation that's off chip. We find that very important to make sure that what we saw on the chip is reproducible. Now, I'm not going to show that data for the purpose of time. And then we do genomic characterizations on it to figure out what's there. And so our isolates, as we'll call them, look like they primarily contain Acetobacter, Enterobacter, and Serratia bacteria. So that's the genus that looks like are the promoters of azospirillum. Okay, so now the second part, this was very new to us, which was now taking our synergistic isolates and seeing if they're going to improve plant growth when we pair them with azospirillum. So what we did was we took dry corn seedlings, we sterilized them, to knock out any other microbes that could be there, germinated them, and then inoculated them either with just azospirillum as the control or azospirillum plus the synergistic organisms that we uncovered. We started with a very basic system and probably the most ideal system for looking at plant growth as kind of step one here, because this was new to us and to our lab. And so we did this just in plant culture. So everything else is sterile here. It's in agar that's supplemented with plant growth media. So a far cry from obviously doing it in the field, but our starting point. And so what we saw here was very exciting. After 15 days, we saw a difference between how long these plants, or sorry, plant height, and how much these plants would grow. And so again, not a big difference at day 5, but at day 15, what we saw was that plants that didn't have any inoculation did not grow nearly as much as those that just had azo. So azo is there by itself doing some growth promotion, but when you pair it with some of these synergistic isolates, you're getting accelerated growth. And so that was really exciting to us to basically say, okay, we're kind of discovering now these isolates from the natural ecosystem and showing that we can use them to improve plant growth. And that's kind of where the connection to biofertilizers is. And so again, this is a very idealized system, but the first step in doing this. Well, that led to a hypothesis, and that was that we wanted to test, and that was what we thought, would be the obvious outcome, but we wanted to test this, and that would be that the corn is growing at higher levels because azospirillum is there at higher colonization levels due to the synergistic bacteria that's also there when you co-inoculate it. And so to test that, what we did is we took the roots and we washed them. So again, those roots only had azospirillum or azospirillum plus the synergistic isolates on them. And then we plated them, and we plated them in antibiotic media, which will only recover azospirillum. So then we can actually quantify the colonization through simple plating. And what we saw here was that indeed, azospirillum was able to colonize and survive on those roots at higher levels when you put it with its friends, with its synergistic isolates. And so we are seeing that synergy translate onto the root and thereby improve plant growth, which again, we were excited to see, as that was really one of the goals of the project. Okay. So plant growth improved metrics, and we believe that's because of the higher level of azospirillum that colonizes on these roots when it's paired with synergistic isolates. So we also thought it was important to maybe do external validation in a little more complicated system. And so we had a collaboration with Dr. Wagner and her lab at the University of Kansas. So they do a lot of plant growth studies themselves with corn in a slightly more complicated system. So this is not greenhouse level studies yet, but looking at corn growth in more of a soil-based system. Here, this is calcium clay. And we wanted them to test our isolates for us, and they also were able to show that you would get some improved growth metrics when you're co-inoculating. And so again, if you look at the data here, the light blue is with no inoculum, and overall, the seedlings on average that were co-inoculated had the highest gains in root and shoot weight. Some were more significant. Some were not significant, but there were some significant differences that we were able to find. That for us really served as a nice external validation that the interactions that we were finding and that we would see improved plant phenotype in our very idealized system were also showing up in a little more complicated system. Now, of course, taking this to the greenhouse and to the field, right? Those are all steps that we have not done, but would need to be done to further progress it. But we're really excited about this preliminary data. So project outcomes and future work. What we showed was that root samples can be screened rapidly with our technology we call the MRA. We can rapidly identify synergistic bacteria that are going to help an existing plant growth promoting rhizosphere survive and grow in a root environment. And so we saw that that also translated to improved plant phenotypes as measured by different growth metrics. So those were our main outcomes again, and that was really something that we know will help us a lot moving forward. Our ongoing work, we're still working to fully characterize our genomically characterize our synergistic communities, and so that there's been a little bit of complication there, but we think we're getting closer to having that wrapped up, and that will lead to our manuscript, which we'll have hopefully submitted in the near future. A portion of the data has also been included as preliminary results in a DOE proposal. So it is helping us move forward in areas. And so I'd like to acknowledge the people that were involved here, specifically my graduate student, Naloy Barua, who is now a postdoc at the University of Florida, for his work, as well as K-State agronomy, Dr. Ruth Diaz, for helping us sample, Dr. Alexander at University of Tennessee for the Azo strain, and then definitely Dr. Wagner and her student, Kayla, for their work. And then also thanks to Bayer, the biologics R&D group at Bayer Crop Science for their interest in the work. Okay, so I'm happy to answer any questions, as well as Dr. Platt. I know he's on the call, and so our emails are at the lower left hand corner of this slide. Thank you.